Personalized medicine is a major goal of current cancer research. The idea is simple. Not all cancers are the same on the molecular level, and they won't all respond to the same treatment. So what we want are therapies that target the unique vulnerabilities of each cancer. For example, say the 10 women on this screen all have breast cancer. Their disease may appear similar, but there are unique differences that we can already exploit for better therapy. 6 out of these 10 women will have tumors that express a hormone receptor, and that might respond to hormone-targeted therapy. Another 2 will have tumors that overexpress a protein called HER2, and that can be inhibited by a targeted therapy called Herceptin. However, the last 2 women will have a type of breast cancer called triple-negative or basal breast cancer. Those 2 women will have worse prognosis, and there are currently no options for targeted therapy for their tumors. And the options are even more limited for other types of cancer. So what's holding us back? The fact is, for most patients, we don't know what to target. Cancers differ from normal tissues in numerous and complex ways, and while we can sequence genomes and use other technologies, we don't always know what the differences mean or which ones are important. Better understanding of what really drives cancer at the molecular level is a critical prerequisite for developing targeted therapies and for personalized medicine. That's where my research lies. I've focused my studies on a family of proteins called R-spondins. When cells make R-spondins, they turn on an important cellular circuit called the Wnt pathway. Wnt promotes cell growth. During normal development, Wnt is necessary for the formation of many tissues. However, cancers can hijack the Wnt pathway in order to promote their own unregulated growth. Earlier work in my thesis lab found that R-spondins might promote colon cancer. In my thesis research, I also discovered that R-spondins are highly expressed by some breast and liver cancers. These cancers show an activated Wnt pathway, and importantly, from a clinical perspective, the breast cancers that express a high level of R-spondin tend to be the basal type that I mentioned to you earlier, has poor prognosis and few treatment options. So how do I know that R-spondins could be a good drug target for these tumors? I've taken basal breast cancer cells that express a high level of R-spondin, and I've experimentally turned off the R-spondin to mimic a targeted therapy. In response, the Wnt pathway is decreased, and those cancer cells slow their growth. These studies suggest that R-spondin could be a good target for drug therapy and provide a new avenue for personalized medicine for multiple types of cancer. Thank you.